So today is part 2 of organelles and their structure and their function. And most of these that we'll be going over today, you're much more common and more familiar with. The nucleus, please get over saying that it's the brain center of the cell. Instead, I would indicate that that is where DNA is located. In fact, if you look at the textbook for the course, it'll label the nucleus as being part of including the nucleolus, chromatin, and the nuclear envelope. So the best thing I would say here is this is where the DNA is located. The nucleolus is inside of that nucleus, and it is non-membrane. It is involved in making ribosomes. And where you've been taught that all eukaryotic cells are membrane bound, remember that in this case, the nuclear membrane is what takes care of the nucleolus. So again, it's where we're going to find RNA, and again, it has function in making the ribosomes. The cytoskeleton would have been mentioned during your previous course, but having said that, now it's going to be taking on a little bit more important of a role. We discussed how it was important with keeping the cell itself, its shape, and inside of it, including in the cytoskeleton, are microfilaments and microtubules. And the microtubules we talked about as far as cell division, that they become more visible during cell division and they are mobile. So the cytoskeleton, again, is for helping retain the cell shape and has microfilaments in it and the microtubules have function in cell division. The plasma membrane we're going to spend an entire class at least on, and that is what has the heads and the tails, the hydrophilic and the hydrophobic parts of all of the membranes. And remember, for example, we've learned that the nucleolus is non-membrane bound. We always think of that plasma membrane as surrounding the cell, and it does. But having said that, it also surrounds ribosomes, the mitochondria, the vacuoles, all would have that plasma membrane surrounding it. Peroxisomes are something we would have mentioned in the college prep course, the honors college prep course. And this is an organelle that it's pretty on the strange side. So it makes hydrogen peroxide, H2O2. And so that H2O2 we're familiar with putting on cuts, etc. I always tell students in anatomy, hydrogen peroxide is actually not an antibacterial agent. It does not kill bacteria. What it does do, though, is removes the oxygen from that area of the wound, and most bacteria the more common bacteria that would be on your skin are going to need that oxygen for growth. So it in itself is not an antibacterial agent. It doesn't kill the germs that would be in your cut. What it does is it removes the available oxygen for it. So inside the peroxisomes, hydrogen peroxide is made, and it's a byproduct of breaking down water, and so it removes that hydrogen. And it also can break down fatty acids in the liver. It helps to detoxify alcohol. We've already talked about lysosomes as helping in detoxifying, but here we now have peroxisomes in the liver that also do that same detoxifying, and it predominantly works on alcohol. And of course, alcohol has a great deal of hydrogen in it. The nuclear envelope is a better term for the membrane that surrounds the nucleus. So we would call this we have the nucleus, the nucleolus would be inside of this, and then we have a plasma membrane or the nuclear membrane around this. But the nuclear envelope is actually a little bit better term to use for that membrane that's around that. Especially in cell division, we talk about the nuclear envelope breaking apart. The chromatin is a term that is used in cell division more than anywhere else. So that chromatin was labeled because it was the first organelle that was stainable. If you reflect back to the onion lab that we did last year, where you took onion skin and pieces of onion and then stained them with iodine, and the rusty brown little circles that would show up where your nucleus is. Well, what's happening is that's the stainable portion of that. And in chromatin, there is both DNA and proteins in that, and that is what causes it to be able to be stainable. And that chromatin, or that stainable component, is more visible just during the early stages of cell division. And so that's where you're going to see the term chromatin mostly is for when we're talking about interphase and prophase. Centrosomes is another term that is used when we are talking about cell division. So here in the cytoskeleton, we talked about the microtubules. Well, during cell division, those microtubules get to be important in that we have these gear-like shapes, the centrioles inside of the cell. And centrioles are these tiny little circles. 2, 3, 4, 6, 7, 8, 9. And last year in my class, I had Tristan Arsha do a nice picture of this because he's much more artistic than I am. And so there are 2 centrioles. And I don't have the centrioles up here, but centrioles would be another one that we could put up, that they are gear-like objects, and they are used in the centrioles and the microtubules attached to each other. And the zone where they're attaching, sort of the area around the centrioles, is called the centrosomes. It's an area. And it's near the nucleus, and inside of that area, again, would be our 2 centrioles, the gear-like objects that are used in cell division. And then, of course, the microtubules are going to be attaching to this. Microtubules that are from that cytoskeleton. And flagella, we don't talk about a lot. It is oftentimes when we talk about bacteria, flagella is going to be the way that that moves. So it gets to be very important there. But we also have flagella, for example, in a eukaryotic animal cell for the sperm. Otherwise, they don't have any mobility. So we talk about that flagella mostly for mobility. So if this was a cell, here would be a flagella off of that. The plastodesmata is for plant cells. So if I can quickly make a nice little, I have tendonitis in my elbow there, excuse me. If I make a plant cell, and remember that the plant cell has the exterior cell wall and an interior cell membrane. Animal cells do not have that cell wall. So here I have one plant cell, exterior cell wall, interior of that would be a cell membrane. And here I have my neighboring plant cell, again with the same sort of structure. Well, the plastodesmata is how they are attached to each other, anchored together. It's a channel, and I've drawn these quite far apart, and actually they would be very close together. It would be something more like this. And they are responsible for attaching and holding those cells together. And in an animal cell, that area of where the cells are attached gets to be very important, because some of the questions that a cell is supposed to ask itself before it divides is there a need, is there room, and is it anchored? Because if it's not anchored, and that's what the plastodesmata does, these channels in a plant cell, and remember now this is very specific, you should recognize the very hard structure of a plant cell. So this is plant cell, as having that cell wall that gives it so much structure. If an animal cell is not anchored, just like this would be, then what happens if there's an abnormality in that cell, if it's cancerous and it breaks off and it travels through the interstitial spaces or the spaces around organs, then that is how it metastasizes the cancer. So a cell that is abnormal, that is not well anchored, could break off from lung tissue where there's lung cancer. That goes through the spaces of the body and flows, and then all of a sudden the person has spinal cancer and brain cancer and liver cancer. So it is one of those important factors. And again, if you remember from the last Screencastify, the lysosomes, one of its 3 jobs is to cell death. So a lysosome should recognize that the cell doesn't belong or the cell is abnormal and should kill it. And it is believed that on any given day, every single one of us have abnormal cells that our body is taking care of, is getting rid of them, killing them, identifying that they are abnormal. So again, to go over these, please don't say brain center of your cell. In fact, I won't give you credit for that anymore. Please talk about it as being where DNA is located. The nucleolus is non-membrane bound, and it is involved in making ribosomes. It's where we are going to find RNA. The cytoskeleton gives the cell structure. It gives it, helps with cell shape. It has microfilaments and the microtubules as both components of that cytoskeleton. The plasma membrane is what controls through channel proteins and ion-gated channel proteins what comes in and out of a cell. And remember, the parts of that are the heads, the tails, again, the proteins that are go through that plasma membrane that allow things in and out. And on top of that would be glycoproteins and glycolipids. So we're going to spend quite a bit of time with that plasma membrane. Peroxisomes are an unusual organelle, and they make hydrogen peroxide as a byproduct, and it breaks down water. It is used when peroxisomes are used in cellular respiration. They are also used to break down fatty acids, and they are used inside the liver. We'll have quite a few peroxisomes in the liver to detoxify alcohol, especially if the person is an alcoholic. They would have developed an enormous amount of peroxisomes within those cells. The nuclear envelope is a better term for the surrounding around the nucleus, that nuclear membrane. Nuclear envelope is a better term. And we say it's a better term because that's what is used specifically when we talk about cell division, that the nuclear envelope starts breaking apart. The chromatin is the nucleus, the nucleolus, and it is the stainable part. It's very visible, the DNA and the proteins, just prior to and during, just prior to cell division and interphase. And the centrosomes are that area that contains the centrioles in cell division. So it's more of a zone just outside of the nucleus. The flagella is the tail-like object that would be on sperm, or how most bacteria move. The plastodesmata are the channels in between two plant cells where they are held together, sort of like a bridging effect goes on. So they're very important. And the centrioles, again, the two gear-like objects that we will find at both ends of the cell that attach to microtubules, and the area around that is the centrosome. And we will spend, on our next class, we will spend a great deal of time on the plasma membrane. Thank you.